And all right, there we go.
Great. Thanks to the organizers for inviting me to speak here today. My name is Elyssa Margolis. I'm a professor in the neurology department here at UCSF. I'm in the division of alcohol and addiction research group. And today I'm going to tell you about a brain circuit that controls pain relief without reward. And just as a disclaimer, as a change from the last talk, this is all basic science. We study circuits and how drugs control the circuits in rodents. So at the center of this neural circuit that I'm going to talk to you about today is the lateral habenula. We've been really excited about the lateral habenula because in a way the neural activity there is pretty simple. The neurons tend to fire more when acute or long-term aversive things happen, such as in this example in vivo recording from a rat that got a tail pinch. Also, the neurons tend to fire less when there's relief to that ongoing aversive condition. And pretty much all of the different glutamatergic inputs that have been isolated and stimulated that innervates the lateral habenula, they all drive aversion. So it's very consistent observation that more activity is bad and less activity is good, which advance.
White hour.
Yeah, no, it's thinking. All right.
Okay.
So just to summarize that idea, these neurons fire relatively slowly in control conditions. When something bad happens or there's an aversive state like a depression-like behavior or anxiety, these neurons tend to fire more in a more kind of ongoing way. And if we can get those neurons to quiet down, either with a pharmacological manipulation or an optogenetic or chemogenetic manipulation, which Tony alluded to in his talk, the animal shows us that it's experienced relief of that ongoing aversive state. The habenula is actually a very small and evolutionarily conserved brain region. This is a coronal section of a rodent brain. It's divided up into the medial habenula and the lateral habenula, which is mostly identified by the very dense set of cells in the medial habenula and more average kind of diffuse level of neuron distribution in the lateral habenula. And we've known for a long time that there is some mu-opioid receptor expression that's relatively highly concentrated in the habenula, but there's been some controversy in the field about whether it's really in the medial or lateral habenula or both. And the reason that looking at this carefully, I think, is very important is that even though these two brain regions are right next to each other and are considered part of the epithalamus together, their neural circuits connect to completely different parts of the brain. So the inputs to the medial habenula are very different from the inputs to the lateral habenula. Also, the projections out of these two brain regions are very different. So for instance, the medial habenula almost exclusively projects to the interpeduncular nucleus in the midbrain, where the lateral habenula projects to midbrain regions that are more involved in mood and reward, such as the ventral tegmental area and the raphe nuclei. So one of the things that we've been working on is trying to very carefully characterize the anatomical distribution of mu-opioid receptors in the BTA to resolve this question of whether the mu receptor is expressed medially, laterally, or both. We're using several techniques. One is immunocytochemistry. We're using a commercially available antibody that's been validated in mu receptor knockout mice. This is an example image where red is nu-n, so each red spot is a neuron in order to more carefully demarcate medial versus lateral habenula. And in turquoise, you can see our mu-opioid receptor expression. And so we see a density of mu receptor expression in the lateral aspect of the medial habenula, but we also see quite a bit of expression in the lateral habenula. We're carefully summarizing this across animals, aligning this border. And so we're looking at the anterior to posterior extent of this brain region because it is a relatively long brain region in the AP axis. And so in the bottom, you can see heat maps averaged across 6 animals, males and females, where we've carefully mapped this out and aligned this medial-lateral border. And a really interesting pattern has emerged in that if we look at the very medial aspects of the habenula, the mu receptor is more highly expressed in the medial habenula. But as you move more posteriorly, we see quite a bit of expression in the lateral habenula, as well as the border right between the medial and lateral habenula. We're also looking at message, so mRNA expression for the mu opioid receptor in medial versus lateral habenula. And while there are a lot of neurons in the medial habenula that have very high expression of mRNA for mu, we have quite a few neurons that are scattered throughout the lateral habenula as well that have lower concentrations of message, but it does appear to be there. So with that anatomical information in mind, I'm going to move to more functional studies where we've looked at how mu opioid receptors control these circuits and how they contribute to pain relief. And in the last minute, I'll show you just a teaser of some data that we have on ketamine because it's another system that we're looking at in a neuromodulatory kind of way. So the model that we used for this work is a spared nerve injury in rats. And we're measuring the behavioral responses to our manipulations of mu receptors, specifically in the lateral habenula with conditioned place preference, as well as peripheral sensory testing. When we microinject a mu agonist directly into the lateral habenula, on the left, you can see that in sham animals, so they don't have ongoing pain, we don't see a place preference. They don't seem to care about the mu activation in the lateral habenula. But once they have the ongoing pain in the SNI model, the mu receptor activation in the lateral habenula produces approach, and we're interpreting that as relief of the affective component of the ongoing pain. In the von Frey fiber tests, we see that, again, in sham animals, there's no impact of mu activation in the lateral habenula, but in animals that have the spared nerve injury, we see a reversal of the hypersensitivity to this mechanical test. In females, we see a slightly different pattern. We have to go up to a higher concentration of the mu agonist DAMGO in order to get the conditioned place preference. That's the middle graph. And we don't see a change in the sensory testing. So we see an impact on the affective component of pain here, but not the sensory. So how does the signal get to the lateral habenula to signal pain and also to be modified by mu activation to get relief? Our hypothesis is that it's coming in through a glutamatergic input because, as I said earlier, glutamatergic inputs to the lateral habenula when they're active are aversive. So we wanted to figure out if there was a single input that we could identify that was carrying this pain signal. And we figured it was probably one that was inhibited by mu, since mu is having this behavioral effect in vivo. We isolated different glutamatergic inputs to the lateral habenula using optogenetics. This means we can express a light-sensitive channel in specific brain regions and then stimulate their terminals in the lateral habenula. And with slice electrophysiology, we can isolate those glutamatergic inputs and test which ones are responding to bath application of DAMGO in the slice. And even though we looked at a variety of inputs that have been associated with pain and chronic pain signaling, including the anterior cingulate, the ventral pallidum, the lateral hypothalamus, and the VTA, the one that stood out as the most sensitive to mu opioid receptor activation was actually from the lateral preoptic area of the hypothalamus. So we went back into the animal to test if this, in fact, could be modulating the animal's affective response to pain. So here, again, we're doing spared nerve injury. And instead of putting damgo in the lateral habenula, we put an inhibitory opsin into the neurons that project from the lateral preoptic area to the lateral habenula. So when we deliver light to these neurons, we're decreasing the amount of firing. And just like with damgo in the sham animals, when we do this manipulation, we don't see any behavioral response. But in the animals that have the ongoing pain, the SNI, we see approach to the side of the apparatus where the animals receive the decrease in activity in that input to the lateral habenula. So now we have an idea of how that pain signal gets to the lateral habenula, and in ongoing studies, we're looking at how that pain signal gets out of the lateral habenula. Where does it go? So one thing that we noticed with slice electrophysiology experiments early on was that when we found glutamatergic inputs to lateral habenula neurons that were inhibited by mu, those lateral habenula cell bodies were also inhibited by mu. So we have sort of this dual control to very strongly inhibit not only those inputs, but the cell body activity specifically for a circuit. So our hypothesis was, well, maybe, or idea, I should really say, was maybe the pain signal is going out of the neurons that are also somatodendritically expressing the mu receptor. So let's figure out what the projection target is of those mu-sensitive cell bodies in the lateral habenula. And we do this experiment by putting a retrograde tracer into different brain regions and recording from the cell bodies in the lateral habenula. This is an example of a neuron that was labeled by the retrograde tracer DII and filled during the recording with a marker biocitin. And this neuron was hyperpolarized by bath application of damgo in the slice. This neuron actually projects to the parabrachial nucleus, which we thought was a really interesting candidate, given that compared to many of the other projections of the lateral habenula, is kind of directly getting into the pain circuit, which is what we would hypothesize is necessary in order to get the relief of both the affective and the sensory component of pain that I showed you early on. And I'll just compare this to another projection that we're looking at, which is the lateral dorsal tegmentum. And while we do see some inhibitions in the neurons that project to this brain region, the projections to the parabrachial are much more consistently inhibited by mu activation. So the circuit that we're looking at here is that we think the pain signal is coming into the lateral habenula from the lateral preoptic area. Mu opioid receptors can dampen that signal by decreasing the glutamatergic release from those terminals, as well as postsynaptically inhibiting the lateral habenula cell bodies that are receiving that input. And those cell bodies probably project to the parabrachial nucleus. And we think that this is exciting because not only is this telling us a part of the brain circuit where you can get this relief without reward under control conditions, but maybe these are circuits that we can modulate with alternatives to opioids that would be safer. And so in the last couple of minutes, I'm going to switch gears and tell you about a neuromodulator, a tiny bit of a project that is really spearheaded by Lakshmi Devi at Mount Sinai, but we've been doing some slice electrophysiology work with her. And so for several years now, she's done a variety of work characterizing ketamine in heterologous systems as a positive allosteric modulator of the opioid receptors, especially the mu opioid receptor. So this means that if you just add ketamine and the receptors don't have an orthosteric ligand, it won't really do anything. But if there is a ligand like morphine or an endogenous opioid peptide, it will enhance the activity of that orthosteric agonist when the ketamine is on board. And so we looked at this in brain slices to see if this is also happening in intact circuits. Here we're looking at the GABAergic inputs to the ventral tegmental area neurons. These GABA terminals are very strongly inhibited by mu-opioid receptors. So it's a really nice place to do some pharmacology and evaluate different ligands. Here we use metencephalin, which is a possible endogenous opioid peptide that might be released in the VTA, to see how ketamine might be working with the peptides that are actually released endogenously, kind of relying on the endogenous system. And so in the example to the left, this is looking at evoked GABAergic synaptic currents. In black, we have the control amplitudes. In red, you have what's the inhibition by metencephalin. So it's maybe a decrease by about 50% amplitude. And the mu receptor antagonist, selective antagonist CTAP, reverses that effect. That's a pretty high concentration of metencephalin. It's 10 micromolar. When you look on the right, we can go down to a lower concentration of metencephalin in the presence of a relatively low concentration of ketamine and get an even larger inhibition of the GABA release in red. And again, it's reversed by the CTAP in gray. So these are the summary data. And I think it's really I was really quite amazed by these results, because in the presence of 10 nanomolar ketamine in our dose response curves on the right, we're increasing not only the potency, so the purple curve is shifted to the left by about an order of magnitude. We're also increasing the efficacy of metencephalin. So the efficacy in orange is kind of a partial agonist at the mu receptor at the specific synapse, even at our highest concentration of 10 micromolar. And ketamine is facilitating that metencephalin response, basically turning it into a full agonist at the synapse. So maybe this is one of the things that ketamine is doing in vivo, kind of modulating the endogenous modulators. So with that, I'd like to thank the people in the lab who helped with this work, as well as our collaborators who have been involved in multiple ways that in these projects that I didn't have a chance to talk about today. I also want to thank the Painless Research Foundation for their support in our research into the lateral habenula, which is some of what I shared with you today.
Thanks, Anita. Question? Yeah.
No. Okay. Very interesting talk. Thank you. And I'm very curious about the upstream. I feel like there's this endless, like, what's upstream of this circuit. And the LPO is a bit surprising to me. I'm a novice in this part of the literature, but as far as I can tell, like a homeostatic region of hypothalamus. Is there a direct input from the nociceptive circuits through LPO, or is this some sort of like contextual physiological modulator of an otherwise separate nociceptive input?
Yeah, so my collaborator at Rutgers, David Barker, is looking very closely at this now. I can tell you that there is in the literature a couple of very old reports that there might be some direct spinal input to the lateral preoptic area. So he's going to check with modern techniques if that is correct.
I think that was really nicely done, seriously. The second thing on ketamine, I have to ask, so is this an NMDA receptor?
So.
Can you repeat the question, please?
Yes, I'm just curious. Ketamine's an NMDA. I mean, it's among other things that it does complicated drugs, but it also has NMDA antagonist effect. I'm just curious if you ran something like MK801 or AP5 or something like that? Did you see something similar, or is it something peculiar to the ketamine story?
Yeah, so we haven't tested other NMDA receptor antagonists in this fashion in our slice work. In Lakshmi's heterologous systems at 10 nanomolar ketamine, she's not hitting any NMDA receptors. They have to go up to about a micromolar to get a direct NMDA receptor response. I think, I'd have to go back and check my notes to tell you if we ran the experiment in AP5. Yeah. Just because it's a synaptic experiment and we don't often do that. Yeah, Mark.
Thank you. Any idea why we see such a range of responses in these models where we have some minimal changes and other, one or two that have a much more dramatic effect on the effect? Does anyone, we tend to just be happy when we see everything kind of going up, but I was just wondering if there's any interest or ways to distinguish other features that would explain why some are seemingly super responders and others are not.
Yeah, so I think this is a great question, and it's something that I think about a lot. One simple possibility in the case of the ketamine is that different inputs to the VTA neurons have different amounts of mu opioid receptors on their terminals. And so in some cases, we can just get a ceiling effect and can't get any more inhibition because the way the experiment is designed, we're not always activating all of the inputs. And so you could look potentially like we did for the inputs to the lateral habenula, maybe at specific inputs and figure out which one has the most ketamine sensitive mu opioid receptors. Is it possible that some mu receptors are more ready to interact with ketamine than others? I think is an amazing biochemistry question, and whether that's related to modifications to the receptor or which signaling pathways they're coupled to, or the exact kind of orientation of the shape of the receptor. Is it a splice variant question? There are lots of different properties of these receptors that I think we're still scratching the surface to understand why some respond more fully than others in different brain regions.
And just the reason behind that question, of course, is clinically we apply ketamine as a supplement or what have you, or a primary. We see quite a range of responses. Some have seemingly no effect, others have a tremendous, and again with minimal side effects, or somewhere in the middle. So I was just wondering if there's a correlate.
Mm-hmm.
Thank you.
There's certainly a range of human responses to opioids too, right? So it's possible. Jared.
Oh, thank you. I'm sorry, we're skipping over that point. So is there any evidence, do you know if it's targeting the same, like ketamine is acting the same site on the receptor or is there evidence to suggest for or against?
Yeah, so if you go to
Elyssa,
very
can
high...
you repeat the question, please?
Oh, yeah, sure. The question is ketamine binding at the same site as an orthosteric ligand? So the evidence that we have so far is that if you go up to very high concentrations, like 100 micromolar or something like that, you can find the ketamine in the orthosteric site. But for the modifications in signaling that we're seeing, we think it's binding out at a positive allosteric modulator site outside of the orthosteric site. And Sushma Jumdar at WashU has identified, I think, 3 to 5 potential positive allosteric modulator sites on the mu opioid receptor.
And then how do you plan to test this behavior?
Yeah, that's another discussion. Yeah, we can talk about that later. I think.
So we have one question from Peter Bell.
Mhm.
To the extent that you mentioned the relationship between the lateral habenula and the raphe nuclei, would you imagine there might be a contributing influence of serotonin to the effective response / sensitivity to pain.
Yes, I think that's possible. We have time for, yeah, Tony.
I was gonna ask,
Yeah, I wondered, two more.
you know, the pre-optic thing is really interesting because although it's there for other reasons perhaps, but it plays a major role in diurnal cyclicity and thermoregulation. You kind of wonder if this has something to do with, we know there's a well-known cyclicity to pain. Just to throw that out.
Yeah, I think it's a really interesting idea. And David Barker and I are very interested in teasing apart the role of the lateral preoptic area in pain as well. He's found that there's also input from the LPO to the ventral tegmental area that also plays a role in pain. So it isn't just limited to this projection to the lateral habenula.
All right, thanks so much.
Thanks.
All right, next I'd like to introduce myself. Thanks. So my name is Prasad, as many of you know. I'm a neurologist and a pain physician at UCSF. I run a laboratory at UCSF focused on understanding brain mechanisms of pain in the Department of Anesthesia, Neurology, Neurosurgery, and the graduate programs of Neuroscience and Bioengineering. So today I'm going to talk to you briefly about the history of DBS or brain stimulation, specifically DBS for pain, and what our lab has been doing to try to understand brain mechanisms of pain by dovetailing off of clinical trials in humans. These are my disclosures. So it's interesting. We know, or we acknowledge to a large extent, that pain exists in the brain, right? That there are various circuits or various brain regions involved that show kind of increased PET activity and FDG glucose, PET, glucose uptake during thermal experimental pain. In this study, it's particularly interesting. In healthy subjects, they hypnotized folks during thermal pain experiments to believe or experience pain with the same intensity that they might usually feel, but be less bothered by it, right? So they tried to influence the unpleasantness of pain. And here, Kathy Bushnell's group in 1997 is kind of arguing that one area that seemed to distinguish or discriminate the affective dimension of pain was the anterior cingulate cortex. This is really cool. You know, they did this through hypnosis, which is particularly interesting. But I would say one downside of this finding was I think people got carried away with this idea that somehow certain brain regions are specific for certain emotions or experiences or activities. You know, we know that there's no pain center in the brain. I would say the ACC is not the affective center of pain in the brain, but it's a region that plays a role, right, in the affective dimension, not exclusively. Anyway, you know, about maybe 10, 15 years after that, Tor Wager and colleagues published this neurologic pain signature where they used a whole brain fMRI in healthy subjects again, right, to try to identify if they could come up with a brain signature using fMRI bold activity, right, where activity increases or decreases, again, in response to a thermal pain stimulus, he may predict what number someone's going to report from 0 to 10, right? And so they found using 13 brain regions, some of which were had increased blood flow, and like the DACC, and the other ones that had decreased blood flow, like the precuneus. In an entirely new cohort of subjects, or healthy humans, they could tell what number they were going to report just based on fMRI scans during the task. Another interesting study, you know, around about a decade earlier, identified using PET studies that placebo analgesia, right, analgesia that's induced by expectation, actually seems to overlap with the opioid network, and so using opioid-based PET tracers, they found that two regions in particular that seemed to co-locate with the mu opioid receptor PET ligand with the rostral anterior cingulate cortex, the rostral. Here I'm calling it the OFC, the orbital frontal cortex, but also often referred to as the ventromedial prefrontal cortex. Anyway, okay, so pain is all over the brain, right? It's largely studied in healthy folks, but of course, there are a lot of studies in real-world chronic pain as well. And so probably the best known research on brain imaging for patients with chronic pain has been done by Vanya Perrin's group at Northwestern. And in patients with chronic low back pain, they found that simply looking at resting state, this is kind of interesting, resting state bone activity in the medial prefrontal cortex, also the orbital frontal cortex, they could actually, that's a pretty good correlation, right? They could predict what number the patient was going to report while they were lying in a scanner. And so, it's clear, at least, it started to emerge that the chronification of pain, or the transition of acute pain to chronic pain, doesn't merely involve kind of increased signaling among identical circuits, but probably involves the transition to, involves maladaptive plasticity with rewiring of the circuits involved in acute versus chronic pain. And similarly, in the follow-up study, Vanya Perrin, his group showed basically here you're seeing in the gray bar are people with subacute back pain, right, who remit, right, that's the R. And in black, you're seeing subacute back pain who persist. And what's interesting is that there seemed to be the stronger or tighter connection, increased functional connectivity between this medial prefrontal cortex and the nucleus accumbens, commonly implicated in reward or even in addictive behaviors in patients who had persistent back pain. So there seems to be essentially an interesting overlap, right, between potentially the biology of addiction and substance use disorders and the chronification of chronic pain. So, all right, we're pain doctors, or at least I am, and often, we aim to treat a lot of these pain syndromes based on what we think is causing the pain. Here's a great review article from Pereira and Aziz where they show in red very common lesion techniques, and in green, very common stimulation techniques or things that have been tried over the last century to try to treat pain. I think motor cortex stim is miscolored here, but anyway, so through the entire neuroaxis, there's been attempts to try to relieve pain from various etiologies, recognizing that pain generators certainly exist in the periphery, in the spinal cord, and in the brain. The thinking or the teaching has always been, if you lesion something in the periphery and you start cutting more proximally, eventually, if you treat something in the spinal cord and the pain's still there, it must be coming from the brain, right? And so that's still how we think about it, but I'm not sure. Well, I'll just say, it's still how we think about it. I'm not sure that's the right answer. A lot of our current thinking about the affective dimension of pain comes from these early studies by Foltz and White in 1948. And so, you might have read about cingulotomies, right? And so the idea that lesioning part of the brain could actually relieve pain was first, I think, studied in rats and then cats, and really applied to humans as early as the 40s. And so here you see that they're writing pain relief, right, in quotation marks. And this is really interesting, right? This is the inspiration for Kathy Bushnell's study, nearly 50 years later, that basically, where they identified that if they lesion this area right above the lateral ventricles, patients reported they were still in pain, but it didn't bother them and it didn't actually influence, their daily life. There were some deficits in other patients, but this is why, based on these cingulotomies, that the anterior cingulate cortex and related circuits became kind of implicated in pain affect. So, okay, I've talked to you about lesions, I've talked to you about stimulation, as well as evidence that pain is in the brain. Believe it or not, perhaps the first invasive human brain stimulation experiment was done for actually treating chronic pain. This was in 1960 in France, published by Mazars et al. They experimented in the 58 and 59 with patients with schizophrenia who had comorbid pain disorders. After that, in the United States, the first DBS for pain was actually here at UCSF. done by Hosoguchi and Adams, and later by our very own Howard Fields, right when he joined UCSF. I think he was in his 30s at that time, but this was 1974. And there they dropped electrodes. Believe it or not, this is interesting. So lesioning was much more common back then. Right? So they dropped electrodes into the ventral posterior nucleus of the thalamus. These patients had horrible facial pain. They lesioned these nuclei, or actually ventral posterior medial, right? Because that's the face. Patients got better for a while, right? 6 months, 8 months. The patients then came back and said, "Doc, my face is completely numb, but now the pain has come back." So yeah, they had anesthesia dolorosa. So all right, we're going to drop electrodes back in, and instead of lesioning, now we're going to stimulate. And so they stimulated, and they found that they could actually relieve pain even from anesthesia dolorosa in these anecdotal studies. Since then, the largest trials done by Medtronic and published, summarized really nicely by Rob Levy, show that when you target the most common historical targets, the paraventricular gray or periaqueductal gray, the VPN, the ventral posterior nucleus of the thalamus, some patients, maybe 50%, get benefit up front, at 3 months, but there's significant attrition over time, right? So a lot of patients lose that benefit, especially beyond 1 year's time. To date, there've only been 3 randomized controlled blinded studies looking at DBS and pain. I told you it started in 1960, right? So for the last 65, 70 years, there's only been 3 studies that have been randomized double-blinded. This is one of them, looking at refractory pain from various pain syndromes. And again, really interesting follow-up. Follow-up over at the longest here, 4.9 years. And it's interesting, they essentially, in patients who were implanted, they did quite well in the long term. But the truth was most patients were not permanently implanted, based on inpatient trials. So more recently, the ACC was popular in 1948. There's a renewed interest in 1997. And then in 2017 or 2014, Tipu Aziz and group targeted the ACC again with DBS to treat various refractory chronic pain syndromes. Here in a cohort of about 12 patients, they show that there's 8 out of 10 baseline NRS, which improves to 4.3 at 6 months, but that effect really at a group level goes away after 12 months. So again, some transient benefit maybe. What's really happening, I think, is that there's benefit in some patients, but it's not lasting. It's not lasting, or 2, there's really particular subtypes that we haven't identified yet that are best treated. That trial was not randomized, by the way. So here's a second randomized double-blinded trial done by Andre Machado and Scott Lemke, many of you know. They targeted the ventral striatum and the antral limb of the internal capsule. Negative study, right? 10 patients with post-stroke pain did not help, but they all wanted to keep the device. It made their mood better, and they had better quality of life regardless. And now this is actually a target in experimental trials for major depression, the anterior limb of the internal capsule and the ventral striatum. So interestingly, Lisa, DBS for headache, hypothalamic stimulation is been quite studied for cluster headache, and it remains to be, I would say, one of the more promising targets. Mechanism of action is totally unclear, right? But I want to put that there. I'm really curious about the preoptic target. So our motivation in my lab has been, okay, one, can we identify a manner or an algorithm in which we can provide personalized stimulation through a closed loop controller, much like a thermostat, just delivers heat or air conditioning when necessary. First, we need to identify biomarkers in which we might respond to the pain, right? The assumption was that if something works, but it wears off in the long term, this may be due to excessive electrical dosage, right? If we limit the electrical dosage, or we only stimulate when it's absolutely necessary, perhaps we can avert this kind of tolerance and develop a more durable brain stimulation therapy for folks. So in this study, we recruited 4 patients who had refractory chronic pain. They had been failed by all other therapies. 3 of them had post-stroke pain. 1 had phantom limb pain. All of their pain was unilateral, as seen by these pain body diagrams, where red is very severe. They drew this themselves, and green is mild. These patients were implanted with a novel bidirectional DBS device where we're capable of recording and stimulating. There's many brain targets, right? So how do we decide where to implant? Based on the literature and a desire to do something that was theory or hypothesis based, that hadn't, well, I'll say hadn't been done before, but also hadn't shown failure before. We targeted the orbitofrontal cortex or the ventromedial prefrontal cortex, which we know is very implicated in chronic pain, as well as the anterior cingulate cortex. Here's an X-ray showing an example of one patient with leads. That's the device with the leads there. And you can see a class brain view with all 4 patients, an example of recording on the bottom right. So in this study, we asked patients after implantation to go home and report their pain 3 to 5 times a day. And amazingly, these patients had such severe pain, but their pain did fluctuate quite a bit. And so what's really cool is every time they reported their pain, they could click a button on a remote and capture a 30-second snapshot of their pain, of a brain recording from the anterior cingulate and the OFC. So here we have pain reports, ecologically valid in the real world, simultaneous with brain recordings. So we used various machine learning tools, and I'm going to kind of skip through this quickly. I'm going to go about 2 minutes over, I apologize. But the bottom line is, using signals from either brain region alone, we could actually predict whether that patient was going to be in a dichotomized high versus low pain state. So we couldn't track, the actual pain number, right? But we could tell, okay, classification. Are they going to be in an elevated pain state or not? And we used various brain signature markers, to build these classifier models. And we even compared them, and these are heat maps kind of showing the features involved. Across the row is, various frequency bands. Peter, you're talking about, frequencies being important in medicine. Frequency bands in the brain, in these various brain regions, and each heat map is one per patient. We were only able to decode in 2 patients experimental pain from a heat stimuli, right? And so we found that even when we could decode the experimental pain, it didn't map didn't match the biomarkers for the chronic pain. So this study kind of provided a proof of principle that yes, we can track clinical chronic pain states at home, and it kind of validated the intuition. Chronic pain is not just a more durable version of acute pain, right? There's different circuits involved and different dynamics involved. And so we got kind of an angry letter, actually, after we published this study from some folks in Germany. And they kind of complained, like, why would you want objective markers of pain? Why don't you just believe the patient? And so, well, I'll tell you why in a moment we might want that. But, really, it's not enough, I think, to just believe patients. I think we have to develop better therapies, right? And their concern was patients might you might use these biomarkers to confer distrust. You might say, your biomarker doesn't show up, so you're not really in pain. So, at the end of the day, I have to say we should be very cautious, right? Pain is defined always as someone's report, right? When they report it is the ground truth. And so, in contrary, I think you can provide validation to folks when nothing else, nothing else on an MRI or lab tests, demonstrates that they have pain. So we're currently conducting a clinical trial using an approach with multiple electrodes in the brain. And here you can see stereo EEG or depth electrodes doing a 10-day inpatient trial. So patients come to Parnassus Hospital or are in the hospital for 10 days after these electrodes are inserted in the operating room through stereotactic procedure. My team works with them for about 8 hours a day, every day. We systematically stimulate and record from these electrodes, trying to identify a region that's going to work the best for that patient. If they get pain relief during this trial, they get implanted with a permanent device, and then we follow them at home in this study for 2 years. The goal, again, was to identify individual biomarkers of pain to try to develop a closed-loop brain stimulator. So here's an example of one participant that's been in this trial now for 1,400 days. And here is before any surgery. The inpatient trial, the 10-day trial that I just showed you, his pain starts fluctuating. We even reached a 0 out of 10 on one day. This is a guy with complex regional pain syndrome as well as severe degenerative spinal disease. We implanted the permanent stimulator and didn't turn it on until about day 450, when you could see his average pain is depicted in the red line is basically at been hovering around a 10 out of 100. And so we then completed double-blinded crossover testing. So I want to show you, so I've gone over quite a bit, but I actually think it's worth taking about 2 minutes to show you these 2 videos. All right, so I'm gonna show you this video of what it looks like in the hospital when we're testing, because it's pretty amazing when we discover new targets. Oh, we're not gonna get audio here. I'm worried. Well, in any case, all right. Sorry, the audio is not coming through. Well, so it is. In any case, what we do is, it's kind of a tedious experience, but we're systematically testing various targets. And here we discovered a new target, which is the left caudate, which had been studied in cats before in the 1980s and not really reported, in humans. The same target that's used to treat tinnitus, actually. And so we're writing this up. It's pretty amazing to be in the room when these patients experience, such relief. So to wrap up, I showed you at the very beginning that 10 years ago now, we were able to predict people's actual numerical pain scores using fMRI, right? Using stereo EEG now, and this was experimental pain using fMRI. Using stereo EEG, we can now, by sampling so many brain regions, actually predict the individual numerical pain scores using signals from up to 25 brain regions. And so it's really remarkable. Our classification has gotten much better. We're identifying what circuits are really unique to each individual and which generalize across populations. And here is just an example of a patient's deep brain stimulator, right? So normally in classical deep brain stimulation, you'll have stimulation on all the time. In this case, this gentleman's getting dramatic relief. And here in the x-axis is 7 days, and you're seeing the percent of time that his stimulator is on. That black line shows it turned on just in response to biomarkers that indicate that he's about to enter a high pain state. And at most, his stimulator might be on for 40% of the hour, but really it's turning on and off, in demand. And these states are defined by us, in the lab, really as essentially low pain, high pain, or overstimulation. And so I'm going to skip the video because really without the audio, I think it's hard to appreciate. But thanks for your attention, and I'll take any questions.
Well, just great work, Prasad. It's encouraging to me as a clinician, it gives me optimism. We may zero in a little bit on what's causing this problem. And I think the most important thing you said is we need to get away from the idea that there's a pain center. There's a single pain center. But it adds to the complexity of the problem and getting a tool to meet, because it seems though there are several pain centers that work in contribution within each individual, and then probably over time, even within the individual, the relative contribution to pain changes. So the data needs are tremendous. But where I hope your work leads, and you're a young enough guy that you can get there, is to use probably machine learning or AI to understand the typical, easily obtainable clinical features of patients in the clinic, response to medications, physical examination, mental health issues, and so on, just the clinically available features, and eventually correlate them with the locations of the pain generation, I'll say, in each individual patient. That will be a tremendously useful tool. But the data needs on the front end are just so difficult because of the multiple sites and also the technical challenges of obtaining the data that with your current method, having implanted electrodes. But just fantastic work, and like I say, it gives me optimism that we're heading in a direction that can be really useful to our patients, rather than saying, "Can you tell me where your pain is and just what does it feel like?"
Right.
That's the main informational question that I could use in the clinic today.
Exactly. Well, thank you. Thanks, Eric. The only thing I'll add to that is, I totally agree with you. We need to essentially get clean or usable big data sets, right, that we can analyze to identify things like where's the pain coming from. But Julian Motzkin, one of my colleagues, is going to be talking about some really cool kind of non-invasive biomarkers that might help us to pinpoint all this. And he's going to talk, I think, in about 30 minutes. Oh, 20. Sorry.
I have to ask. So all the patients you showed, it wasn't clear that all of them had an organic source. But maybe most, let's just say all the back pain. And if you were to take those patients, and so you've seen these biomarkers, I'm curious, if you did a spinal block, so do the biomarkers regress to a normal state, or do they persist? Which I think is kind of an interesting question, because I've always wondered what happens if you get intrathecal morphine, not a block, but just knock out part of the pathway that normally leads to, oh, I don't have pain.
That's, you know, sorry, it's funny, like after these electrodes in their brain, right? We basically we do various tasks with them. We try to understand or modify, you know, single variables. We have not yet ever done that. Like we haven't just done like a spinal block or tried to do a peripheral nerve block. Because you're right, so these things help or might abolish their pain for an hour or so. But how might their biomarkers change? I don't know.
But I mean, the point I think is that there's no doubt that the final integrator is up here, and that there are ways to generate the pain-like equivalent secondary to stress. And you don't need the injury. But I guess what I'm getting at is, are these pathways that arise out of the spinal cord secondary to, say, some trauma or whatever, are they regulated in the same way by morphine? I mean, just like the ventral cordotomies.
Yeah.
Though there's not a really strong literature, surprisingly enough, given the numbers that are done. But the data that are out there suggest that when you do that for things like osteosarcoma of the lower extremities, the general response the patient has is, I feel better, I'm not as depressed, etc. Some would say, well, that's just due to the fact you're not painting. But I think it's more than that.
Yeah, that's really interesting, right? Like if we could, well, yes, to what degree do the afferents, right, like play a role, even in perpetuating, you know, non-somatic symptoms? I think that's very interesting. It makes me want to write an IRB for that.
Yeah, but not systemic morphine.
Right, not systemic. Yes, intrathecal
Or epidural.
Sure.
I'm just asking on behalf of Alan. Please go ahead, Alan.
Thank you, Prasad. You know, when I lecture the medical students, one of the first things I tell them is one of the problems is we don't have a biomarker for pain. So I'll keep my fingers crossed. But I do have a couple of questions. The work is remarkable. Two things. One, I don't expect an answer, is there any connection between a patient with painless embolia, where you've got a temporal loss of affect, pain affect, and all of the focus on anterior cingulate? So we'll leave that for a moment, but it's just a question that I've wondered for a long time. The other one is your distinction between acute and chronic pain. If in these patients, first of all, you're talking about ongoing pain versus evoked pain, or and if you now provoke pain, if you could do it legally, in these individuals with a peripheral stimulus, would they experience it even though their ongoing chronic pain is gone?
Right, that's a great question, Alan. And so the short answer is yes. And what we learned, that yes, they can experience various normal, acute, you know, appropriate pain, even though their chronic pain is gone. And that's critical, right? So we had to actually teach patients to actually discriminate between various other pains that they were getting throughout the trial and their usual background pain. We had one patient who had horrible diabetes. His foot was hurting and he felt pain, but it was appropriate because he needed an amputation. He got the amputation, and all of that pain was distinct from his usual pain that he was having. So clearly there's a lot more that we need to understand. We had another patient who had horrible kidney stones. She felt them despite her usual pain.
Any thoughts on the painless embolia?
I have equivalent asymbolia in response to that. No, none. I have thoughts, but it's not worth sharing.
Okay, we'll save it over a drink.
Okay. All right, thanks.
Hello, everyone. My name is Tommaso Di Ianni. I'm an assistant professor here at UCSF in psychiatry. I have a lab in the Weill building, so right across the street. And my lab is really focused on ultrasound. So that's what we do. Many of you know me for the work that we're doing in ultrasound neuromodulation. However, that's not what this talk is about. This talk is about ketamine, and we have been studying ketamine. We're studying ketamine with some of the technologies that we are developing as well. And we have been studying ketamine and the interface of ketamine with opioid receptors and sex differences. And that brings me to the somewhat generic title of my talk, which was intentional, because the way we are approaching this, and this is an overarching goal of my research program, we're really trying to build a bridge. So we're trying to build a bridge between the preclinical work, of which we have seen some beautiful examples from Elyssa, and the clinical work, or the work that we do in humans. So all the technologies that we're using, even for the work that we're doing in rodents, we are always kind of keeping in mind the translational perspective. So when you will see the results, I would invite you to always kind of keep that in kind of the back of your head and try to understand what we're trying to do here. First of all, I would like to thank PARC, Mark, Anna, Prasad, and Lawrence for organizing this. I was also very lucky to receive a PARC Fellowship in 2024, and so some of the work that I'm going to show was actually funded through this fellowship. Okay, so first I would like to introduce functional ultrasound imaging. This is a modality that is very close to functional MRI. So both are based on neurovascular coupling. The nice thing of functional ultrasound, though, is that we get very beautiful sensitivity. So we can see much more than what we can see with fMRI, and I will show some data to make that point. So, but first I wanted to show some videos. This is from the very first paper that kind of introduced this modality. That's a spreading epileptic seizure. So they induce the seizure pharmacologically, and you can see the seizure spreading in, that's a rat brain. So first in the cortex, and then it kind of spreads in the thalamus and in the hippocampus. This other video is from our own work on the right, and it's showing what happens in the brain when we administer a subanesthetic dose of ketamine. So you can see that immediately now we administer ketamine, and there is this surge of activity in many brain regions that we have actually already heard about today. Specifically in, maybe let me pause it here a little bit. So up here, for the ones of you who may not be familiar with the rat brain, oh, it's still going. So up here, this is the cingulate area 1. You don't see the pointer. Okay, I'll make this later. But these are all regions that are very important for many different things. The anterior cingulate cortex, or the homologue of the anterior cingulate cortex, the medial prefrontal cortex, the nucleus accumbens, the retrosplenial cortex, the lateral habenula. So all these regions are showing up in these videos as activated upon ketamine administration. Okay, now I mentioned that functional ultrasound, one of the reasons why we are really excited about this modality is that it is a lot more sensitive than fMRI, because otherwise one may think, why don't just use fMRI? We are interested in building a bridge, however, with fMRI, because whatever we find in rats, then we can possibly more readily translate into humans. So that's the link. These plots here are from a paper that was published by the lab of Jean Lee at Stanford. They do like opto-fMRI, and so they tried opto-FUSI as well. And then they compared the two modalities. So they're driving activity in the motor cortex with optogenetics, and they are recording on the top with fMRI and on the bottom with functional ultrasound. And just I would like you to focus specifically on this plot here, where you can see that, or like that on the right. On top with fMRI, we don't really get any meaningful signal upon stimulation, but with functional ultrasound, we still get some pretty good responses in correspondence of our optogenetic stimuli. So again, speaking to the fact that we have a much higher sensitivity. So we can see, essentially we have a larger effect size when we do the experiments. We can see smaller effects. This is from our work, but there are other studies that have looked at correlates of functional ultrasound imaging with electrophysiology. So we did intracranial EEG, and we essentially, with different doses of ketamine, we saw very similar responses between functional ultrasound imaging and intracranial EEG. So again, kind of, I always get this question, are those signals actually driven by neural activity? Is it all a kind of cardiovascular response? So for what we can tell, we really think that there is kind of a neural basis to the signals that we're recording. Okay, so now coming to ketamine and the way we're using ultrasound imaging, functional ultrasound imaging, to try to understand ketamine a little better, or like how ketamine works. So I don't think I have to introduce ketamine to this crowd. You may all know that it's FDA approved for treatment-resistant depression, but we still don't know exactly how ketamine is working. And importantly, there were a few studies in 2018 and 2019 that raised the question whether specifically the antidepressant effect of ketamine is mediated by opioid receptors. So in this study here on the left, this is from Nolan Williams, Boris Heifetz, Alan Schatzberg, and Caroline Rodriguez at Stanford. And they treated the patients with naltrexone, so an opioid receptor antagonist, before infusing ketamine in patients with treatment-resistant depression. And they found that the antidepressant effect was nearly completely abolished. Now, that's still, as far as I can tell, it's still a matter of, it's a controversial finding. There were other studies, the ones on the right. One is from Tobias Martin at UCSF. The other one is from the group at Yale that have kind of shown that actually that may not be the case. So we don't know exactly what is happening. But our idea was, okay, let's take the same experimental paradigm, let's reverse translate that in rats so that we can map what is happening in the brain, and we can try to understand these mechanisms in the rat brain. So now if we focus, so these are time series of the course, but I would like you to focus on first on the right side, where we see some statistical parametric maps. So these are group level comparisons between the animals that received vehicle plus ketamine, so just saline and then a ketamine infusion, or an infusion of naltrexone before receiving ketamine. So before we give ketamine, we're blocking the opioid receptors. And then we compare and see what happens if we find any differences there. And somewhat surprisingly, we found statistically significant differences in all these regions that we have heard about today. So CG1 is the homologue, you could tell, of the anterior cingulate cortex, the nucleus accumbens, the lateral labanula, the retrosplenial cortex, also important in the mechanism of ketamine, especially for what concerns the dissociative effects. So it all makes sense. Now, the thing that was very surprising to us was that was only the case in male rats. In females, we did not see anything. We looked across different, we did many experiments with also with different kind of different experimental setups. We did not limit ourselves to functional ultrasound imaging. We also looked at PSD95, it is a postsynaptic density protein that is overexpressed in the prefrontal cortex, and we saw the same effect. We also did behavior, and we saw the same. So kind of wherever we looked, we saw these opioid-mediated and sex-dependent effects. Also, this seems to be specific to ketamine. When we replicated this with MK801, there is a more selective NMDA receptor antagonist, as we were talking about earlier. We did not see any differences. So this seems to be something that any differences between the group that was pretreated with vehicle and the group that was pretreated with naltrexone. So kind of that opioid-mediated component in the response to ketamine was not there with MK801. That is a more selective NMDA receptor antagonist. Also, we then took male rats, we removed the gonads, we repeated these, we let testosterone wash out, we repeated these experiments, and the effect was completely gone as well. I'm only showing 3 regions here in the prefrontal cortex, but across the brain and all the regions that we looked at, there was essentially no difference there. Okay, so now more recently, and again, thank you to PARC for supporting us in this work. We've started looking at circuit level mechanisms, or like rather, the work that I've shown until up to this point is more like we're getting a snapshot in the brain of all these regions where we see all these differences. but we don't know exactly where that signal is originating. We don't know how that signal is kind of evolving dynamically over time. And so for that, to start addressing those questions, we started looking at functional connectivity instead. So here, this is just like resting state fMRI. Nothing new on that front. Essentially, the assumption here is that when two regions are oscillating together, there is a functional connectivity involved. If two regions are decoupled or decorrelated, essentially they are not speaking the same language at that specific time. And this is work that was led by Valeria Grasso, postdoc in my lab, and Joseph Tennyson, who is an undergrad. So first, with ketamine alone, we did see a decorrelating effect in most regions where we did see statistically significant effects. So mostly ketamine seems to, well, I don't know, the colors here are it's a little too bright, I guess, but you can look at the bar plot, the error bars on the right side. In most regions or in most ROI pairs, we do see a decorrelating effect acutely after ketamine that then goes back after 40 minutes. And between some ROIs, we actually see an enhancement in those correlations. Then, because we were very interested in the prefrontal cortex in this study, and the reason is that for reasons that I will mention later, I hope I have the time. How are we doing on time, by the way? Okay. So we're very interested in the prefrontal cortex, trying to kind of decouple the potential hubs of kind of driving the therapeutic effect of ketamine and the regions that may be more involved with potential adverse effects. And so that's the reason why we focus specifically on the prefrontal cortex. And so here we are putting a seed in the prefrontal cortex and then correlating that signal to everything else in the brain. And again, top is males, bottom is females. On the left side is the baseline, so it's pretty much the same. We administer ketamine without naltrexone. There is a decorrelating effect in males. In females, something different is happening. And then with naltrexone, basically naltrexone is changing that response to ketamine in males, but in females, vehicle plus naltrexone and vehicle plus naltrexone look nearly identical. Now, what is important to, what I should point out here is that we also, I'm not showing the data, but we also imaged with naltrexone alone, and we did not see anything. So it's not just like naltrexone is kind of pushing the correlations up, ketamine is pushing them down, and then when we average it out, it's somehow canceling out. It seems to be a more subtle interaction between what is happening. I'm gonna maybe skip over this quickly on the next few things, but we look across different measures. So this is what is called also regional homogeneity or the Kendall's coefficient of concordance. The nice thing of this approach is that we don't have a seed, so it's kind of unbiased from that perspective. We're just looking what happens, but it's a measure of local synchrony. And again, we see these differences in the prefrontal cortex, in the striatum, driven by ketamine that go away when we pre-treat with naltrexone. That is the case in males. In females, we don't see much, at least in the prefrontal cortex. We do see something, although not significant, in the striatum. We also looked at dynamic functional connectivity. So essentially here we are looking at kind of faster dynamics, because again, as I mentioned, we were interested in kind of those dynamic effects. And we find that, so we use like unsupervised clustering. I can go into more detail in the Q&A maybe. But essentially we're like, it's suffice to say that we're looking at more dynamic effects on a shorter time scale than the 15 minutes that we're looking at with static functional connectivity that I just showed. And so we see an increase in brain entropy, which is something that it comes also, it kind of correlates with some of the literature that we have with psychedelics and dissociation in general. But we only see that in males, not in females, and it goes away when we also pre-treat the animals with naltrexone. Okay, this is the same, but I wanted to skip over a little bit to again look at the big picture again in terms of like what we're what we're trying to do. And this applies, by the way, also to the work that we're doing with ultrasound neuromodulation. So kind of the framework that we are using is the same. We took some questions, some interesting questions from the human world. We reverse translated that into rodents, where we can do more mechanistic studies. We can do staining. We can induce specific behaviors or give them enough drugs so that they show locomotor sensitization, for example. But all the modalities that we are using kind of facilitate, we believe, that translation back into the human world because of the close link that there is between functional ultrasound imaging and fMRI. So we're using those to create that link, essentially. Quickly, another reason why that is our motivation, or was our initial motivation, to map those regions in the brain and map the effects of ketamine, especially as it relates to opioid receptors in the brain, was that now we have access to modalities that allow us to deliver ketamine on demand in specific spots in the brain. And so this is something that has been now kind of my PI, the lab that I did the postdoc with at Stanford is a pioneer in these modalities, but now other labs have also kind of built on that and expanded that technology. So we have access to methods to deliver ketamine in a targeted way on demand in the brain. And so if we can get that mapping of where in the brain we can unleash most of the therapeutic response, whether that is for depression, pain, OCD, or whatever we're using ketamine for, but somewhat decouple that from the hubs that are more responsible for potential like dependence or even, I wouldn't talk about addiction necessarily, that's a little bit controversial with ketamine, but at least there is evidence that it is creating dependence. And then on that note, one of the regions that we have enough evidence, I think at this point, to make us think that probably we should avoid sending ketamine to, if we can, is the nucleus accumbens. This is a study that we did with the lab of Mike Michaelides at NIDA. So first of all, rats, in this case, this is as ketamine. All the previous work was with racemic ketamine. But what they have shown is that rats do self-administer as ketamine. So they escalate the number of infusions over a period of 16 days. And with that reverse U-shape dose response that is very common, I think, in this behavior. However, the drug dose is very low. And so we wanted to see, we use in this study, we use functional ultrasound imaging to see what happens with these very low doses. Those are very small signals that are very hard to decode in the brain. And so we used 0.5 milligrams per kilo. So we couldn't see anything with 0.25. It was really hard and very noisy, or rather we would have had to use a very large number of animals. And so we went with 0.5 and we found very rather strong responses, opioid-mediated responses in the nucleus accumbens. For the ones of you who are not familiar, again, with rat brain, this here is the nucleus accumbens in the rat brain atlas. This is the core, this is the core, this is the shell of the nucleus accumbens. So we did see these opioid-mediated responses in the nucleus accumbens. So again, probably that's the region that I would avoid. So if you can send ketamine kind of more in a more targeted way. Okay, so with that, I would like to thank my collaborators, funding, PARC again, for the fellowship and for organizing this beautiful symposium. And this is my lab, and thank you all for your attention.
Time for one question or maybe two. Derek?
I mean, yeah, but if anyone else has. So just thinking about, trying to extrapolate, if you were to take someone who's taking ketamine and you were to give them naltrexone, would they only have a negative experience? If you think about the psychedelic aspect of it, because maybe you're getting really rewarding parts.
I mean, to be honest, I don't know the answer to that question because I haven't I haven't tried. Also, so you're asking specifically in terms about the like the perception or like the experience, right? Yeah, that's an interesting question. Yeah, that's something that it would be nice now to test, given the kind of the fact that we do see these, for example, the increase in brain entropy that seem to correlate with.
You could try this with like condition-based preference with the rats.
Yeah, totally. Yeah, and see if they, they do. So it's like Mike Michaelides also. They did show that there is a preference in the condition-based preference assay, but that seems to be the case only with S-ketamine, not with R-ketamine. So with racemic ketamine, might be a little kind of, because it's more or less 50-50%. So it might be like a weaker response in the condition-based preference. But yeah, totally, that is something that would be very interesting to see. Yeah. Thank you.
Thank you very much. Very interesting work. If I could just ask, opiate receptor blockade robustly modulates ketamine-induced neural activity. So what does that exactly mean when you're saying modulates the activity? And what do you think it means clinically, this modulation of the neural activity and the interactivity you spoke about? What does that mean clinically? And also, what do you mean modulates?
So what I mean by modulates is that, or maybe I don't know how far I can go.
I mean, it's increasing functionality.
Oh, no, it's modulating it. That's the so. If you look at the time courses here, so first of all, green one is just naltrexone alone, so it's almost inert, at least in our responses. The blue curves, that's the response that we see when we administer ketamine alone. When we pre-treat with naltrexone, you can see that it suppresses that response. In this case, in these regions, there are other regions, and actually, it's more visible on the other side. There are other regions where actually naltrexone seems to be enhancing the response to ketamine. Now, the first thing that we thought of was also that based on what we know about the lateral albannula, like, oh, cool. So this is all part of a circuit, right? Especially like the lateral albannula, then the locus accumbens and the prefrontal cortex. We thought that in that plot, what it means is that naltrexone is enhancing the response to ketamine in the lateral albannula. But because there is that inversion, we thought, okay, so the lateral albannula it's more responsive when we administer naltrexone, and that causes like a reduction in the response to the prefrontal cortex and in the nucleus accumbens, for example. Except those are also decoupled in time. So that's why we then became more interested in trying to understand better the temporal dynamics of these signals, if that makes sense. But we talk about modulation because it's not, in some regions it is decreasing the response, in some regions it is enhancing the response, and there is also a time component as well. So it's changing and shaping that response. It's not eliminating it or enhancing it.
So in this particular case, opiates enhances ketamine-induced neural activity.
No, here it is.
No,
here,
that's
what
blockade.
this is blockade. Yeah.
If you didn't have blockade and you added opiate, you enhance ketamine-induced neural activity, perhaps.
So you mean if you don't have, if you are co-administering ketamine with an antagonist?
With an agonist.
Well, that's what we're doing. So here we are pre-treating with naltrexone.
The blockade, right?
So we pre-treat here, and then we administer ketamine. And then we're looking at the differences in the neural responses in those two conditions.
Right, but you could have the inverse, is all I'm saying.
You mean inverse, like ketamine with naltrexone, or?
The response in a different location.
I'm not sure I'm following the.
Right. Right. So it enhances. No. Thank you.
In some other, so in the red regions, it's enhancing. In the blue regions, it's suppressing. Yes, but that's different time points also.
Oh yeah.
Yeah. So you can see, I'm not showing all the time courses here, but actually in the paper there are all the time series in all the regions, and you can actually see what is happening with the different conditions.
Just a quick question. So there's at least two isomers of ketamine. I'm just curious, and I realize most ketamine clinically is, I think, the racemic.
No, the FDA approved this as ketamine.
Is that right? So,
Yeah.
So what have you compared the two isomers?
So this work is all racemic ketamine. The work that I showed in the last slide, that's S-ketamine. And it somehow, we see equivalent responses in terms of like,
Yes.
I mean, so it's different doses and it's different study designs. Mike Michaelides and NIDA, they have done a more thorough comparison between R and S-ketamine. And they found in a study that precedes the study that we collaborated on, and they found that S-ketamine seems to be, like they showed self-administration, they show condition-based preference with S-ketamine, but the response was a lot weaker with R-ketamine. But it was just looking at these kind of addiction-related kind of behavioral parameters. It was not looking at, for example, the antidepressant effect. So that's a different
And then to
story.
get into the weeds, there's two isomers. Of course, the opiates have multiple isomers.
Right, yeah.
So now, so I'm just curious. I know that it may seem pretty trivial, but there are some who argue that there's a toll-like receptor effect.
Yeah, no, totally. I mean, that's
So I'm
possible.
curious
Yeah.
if you did this. I know.
No, we only use naltrexone, yeah, because we wanted to keep that link with whatever had been done in humans. So we kind of reverse translated pretty much what they did in humans in rats to see what happens. But it's definitely it's a very interesting,
I mean, it's
interesting.
almost be surprised if you saw something, but it's just one of those things that.
Yeah, also naltrexone is non-selective to mu-opioid receptors. For other reasons, we do think that is mostly a mu-opioid receptor mediated response from, like, again, that study also with NIDA and so on. But naltrexone itself is not selective to mu-opioid receptors. There is a component also of kappa.
I'm not so much thinking about those. I'm just thinking about, is this an opioid receptor mediated effect? And if it is, some are, that's the whole principle of the original opiate binding work.
Yeah.
Cited.
Yeah, we do show binding. We did other radiography as well, and so kind of all those other studies to show binding and activation actually also of the receptors throughout these regions. Yeah. Okay. Thank you all for your questions.
All right. So we are running 5 minutes behind, which is totally acceptable because I'm to blame. Yeah. Yes. Let's try this. Sorry, Motzkin. Okay. Sorry, let's try that. I'm gonna unshare for a moment. I see. So this is like over here, and then I'm gonna go ahead and share again. Yeah, exactly. I know. I'll try to troubleshoot it for you. You have videos? All right, so we're running 5 minutes behind. Yeah, it's my fault. I delayed everyone. It's our new schedule.
Sorry. Thanks, Prasad. And thanks everybody for coming and for these wonderful talks preceding mine. I'll try to be a little quicker today than I might have been. But so my name is Julian Motzkin. I'm one of the neurology faculty, assistant professor in neurology. I see patients in the Pain Management Center. I had the great fortune to do a postdoc with Alan Bosbaum, unfortunately during the pandemic, and started working very closely with Prasad and had an opportunity to start working on TMS, or non-invasive brain stimulation for pain. And that's really turned into a whole area of research for me, and I'm going to dive into that today. So ultimately, what I want to talk about today is how we use brain stimulation to better understand these transitions from a relief state to a pain state, persistence of a pain state, and then ideally back towards a relief state. So I'm going to briefly introduce pain circuits. Prasad's done a really nice job of doing that for me already. Talk a little bit about stimulation biomarkers, the state of the field currently, and about some new work that we're starting to do, trying to understand this and expand it into a clinical population in which we really don't understand the fundamental mechanisms, and hopefully use these biomarkers to better understand mechanisms, but also improve treatment. So dating back to Descartes, we've been, and probably long before that even, there's been a curiosity about the systems and mechanisms through which the human body can ascertain threat or pain and then respond to that appropriately. He presumed that there may have been a labeled line between the pain sensing system and the pain action system. This idea was formalized in the gate control theory in 1965. It still holds a lot of water and is a great kind of model for us to understand the peripheral and spinal mechanisms of neural transmission in context of pain, and also the influence that higher level circuits, brainstem, has over this process. So including work by David Julius here, we've really understood a lot about the peripheral mechanisms, the central mechanisms in the spinal cord. Even at the time that this theory was first proposed, were reasonably well understood in terms of the architecture, but now we're starting to better understand the dynamics of the spinal cord circuits, thanks to new work with optogenetics in live, freely moving animals. But at this time, and even as this hypothesis was starting to come about, there was a real lack of understanding of what was going on in the brain or higher level processing that could influence these lower level circuits. But with the advent of non-invasive brain imaging strategies like PET and fMRI, and using experimental pain primarily in humans, we start to see that there's a constellation of brain regions that's active in the state of pain. And interestingly, these are brain regions that are not just sensory processing regions, as we've heard earlier. A lot of these regions are emotion processing regions. And Prasad already talked a little bit about the neural pain signature, but it's really a network of brain regions that are involved in different aspects of pain perception. So the sensory processing, the emotional processing, and they're weighted sum of activity together, some that predict positively, some predict negatively, meaning when their activity goes up, pain is higher, or activity goes down, pain is higher, can very reliably predict pain. But this is, again, in experimental pain models. We have started to use this information about the neural circuits of pain in humans and in animals, track tracing studies, and even before some of the more recent fMRI work. Dr. Fields put together a really beautiful summary of pain, sensation, and unpleasantness, and how it interacts in the brain. And importantly, and the reason I bring this up, is there is a very early divergence, as we heard from Dr. Yaksh earlier, in the medial and lateral pain projecting pathways between the algosity, or the sort of nociceptive qualities of pain, and unpleasantness. So that can happen very quickly from the inciting stimulus. But then there are these important higher level functional roles of other brain areas that are involved in the suffering component of pain, meanings that we make of pain, and really the core pathology of chronic pain that our patients suffer with. There's also important circuits that we've heard about today through which the brain can influence pain transmission at lower levels of the hierarchy. We recently summarized the history of deep brain stimulation in the context of these central brain circuits to try to better understand what we know and what we don't know about the architecture within the brain. And ultimately, this is adapted from others who have done this before us. And we know that there are brain regions, again, that are involved in the sensory processing in yellow, the emotional processing, or the motivational qualities of pain in red, and then these more higher order cognitive self-referential processing in blue. And then there are these special regions that seem to be very important for the top-down modulation of incoming pain signals at the level of the spinal cord. We've more recently begun to formalize how do we understand the way that these brain areas talk to each other in the context of pain. And this idea that there's a few different pathways to pain. There's nociception, so strong incoming drive. There's deficient modulation. But then there's also a really important piece of network synchrony, these areas co-activating at the same time that lead to the conscious experience of pain. And Dr. Di Ianni, I think, presented some really interesting data that even speaks to this, that in the pain state in the functional connectivity data that you showed, there's one of the states in the pre-ketamine was a whole brain was highly correlated. And I think that's kind of speaks to this idea that synchrony may be related to the pain perception. But I'm not really going to talk about that. So again, a lot of this we've learned through the experience of acute pain in healthy volunteers, generally college kids. In chronic pain, there's a very limited set of studies that have been done to understand what is the true mechanism of chronic pain in humans. And this is just a meta-analysis of the areas that go up relative to healthy adults or the areas that go down. And there's a lot of overlap between regions that go up and down in different studies, different populations. And when you look at a statistical map comparing these ups and downs, it's empty, which is why I'm not showing it to you here. There's no consistent differences. But in individual studies, we do see that chronic pain is associated with structural and functional changes in emotion processing regions. And some of these you may recognize from Prasad's earlier talk. It's really emotion regions that seem to be most predictive of the transition of chronic pain. So what do we know from this prior literature? We know a whole lot about the architecture of the system, how nociceptive signals are transmitted into the spinal cord and then projected up into the brain, and how different brain areas process those incoming inputs in different ways and then work together to create an experience of pain. But the mechanisms fundamentally in humans of chronic pain remain unclear. So we understand some of the where of pain, but very little about the who, why, and how of chronic pain. So the sort of dynamics that underlie the structure. And so my goal here is to try to understand the fundamental mechanisms of pain through stimulation. And this speaks directly to the idea of biomarkers. And Prasad brought this up earlier, but ultimately, there are no biomarkers that we can use to understand pain. And so the lack of a measurable biological signature of pain associated with diagnosis leads to a variety of things. So it leads to delays in diagnosis, leads to enhanced stigma, especially when we can't see any structural changes like we might see with a herniated disc that causes pain in a dermatome. In fibromyalgia, we don't really have anything to measure in that way. Leads to poor disease classification or biotypes in terms of pain research and mechanistic understanding, and ultimately poor outcomes for patients. And so in fibromyalgia, there are very few patients that have an early diagnosis, and delays in diagnosis lead to increases in symptoms across all of the different types of symptoms. And so I'm going to go kind of quickly through this, but there are lots of different types of biomarkers. So I talked about diagnostic ones, but there are biomarkers for predicting treatment, predicting predispositions to pain, recovery from pain, treatment responsiveness or failure. And so even beyond that, biomarkers can help us do better clinical trials, help us understand pain in individuals who can't communicate, among a variety of other things. And so where are we with biomarkers now? There are a number of candidates, none of which, again, have proven to be effective or reliable in large prospective studies. And TMS may fit at this higher level of understanding brain function, and I'll talk a little bit about that. So very briefly, TMS is a method that uses rapid discharge of electricity through a coil of wire to generate a magnetic field. That magnetic field in turn generates an electrical current in the brain, and that all happens over the course of milliseconds. And the field that's generated is greater than 2 Tesla, so about the strength of a diagnostic magnet, and it induces current at a high degree and neural activity very quickly. A lot of what we understand about TMS in the context of pain, but also from a research standpoint, relates to stimulating the motor cortex, because we can get a readout of what that stimulation is doing to the nervous system. And that readout is when we stimulate motor cortex, a volley goes down through the corticospinal tract and the primary motor neurons, and then elicits a measurable motor response in the periphery. When we stimulate repetitively at a low rate, that motor response over time stays pretty consistent. When we stimulate at a high rate, that motor response gets progressively stronger. And if we do a conditioning pulse before and then after a low frequency train of stimuli, we get an inhibition, so a depression of that response. And with high frequency, we get excitation or enhancement of that response, so kind of LTP-like. So motor cortex is also very important because it's the area that's affected for pain. There's some debate about that, so we're doing a clinical trial. I just kind of wanted to show this. It's very exciting. We're seeing 70% responders in our motor cortex TMS trial for facial pain. But today I'm going to talk again more about the biomarker side. 5 minutes to cover a lot. So the first kind of veridical TMS biomarker that came out was this idea of paired pulse stimulation. So if you stimulate with TMS and then you stimulate at a very rapid rate afterwards, there's inhibition of the subsequent response. You see this drop. And if you stimulate at a longer interval, you can get facilitation. We know that the short interval inhibition is GABA A mediated, and the long interval facilitation is NMDA and AMPA mediated. In individuals with chronic neuropathic pain, that short interval inhibition is reduced. So we get less of an inhibition, so more excitability in the context of chronic pain. 10 hertz TMS, so the sort of TMS that can make pain better, enhances activity, but notably enhances inhibition. So the type of activity that's enhanced, it seems to be GABAergic and limiting the hyperexcitability phenotype. And for people that get pain relief, the pain relief is directly from TMS, is directly predicted by the correction or the rescue of that deficient intracortical inhibition. So speaking to hyperexcitability in the context of pain. More recently, folks have started to look in the opposite direction, how stimulating the brain relates to motor activity in the body. And this also relates to an important phenomenon that we as pain practitioners see, which is individuals in chronic pain stop moving. So there's a really important link between motor function and sensory function in pain. And David Siminowitz, among others, has really pioneered this idea of understanding corticomotor excitability as the amount of brain that you can stimulate that activates the muscle. So if you can move your TMS probe around and get the same muscle activating, that's an excitable muscle. If there's a more narrow area that you stimulate to activate, and as you go outside of that area, you can't activate the muscle, that's a less excitable muscle. So they've done this in a variety of muscles, and they found in clinical populations, subacute back pain, that you get a reduction. So patients with subacute back pain have a smaller area of stimulation, so less corticomotor excitability. In persistent patellofemoral pain, so knee pain associated with musculoskeletal dysfunction, we see a similar reduction in corticomotor excitability in the vastus muscles, so the thigh muscles associated with knee movement. The corollary to that is with TMS at 5 hertz, so an excitation frequency. This is now, instead of just looking at how much brain you stimulate to get a single muscle, this is how much muscle you get from stimulating a single brain. When you do TMS at 5 hertz, there's you've got to move around to stimulate from hand up to the deltoid, but after a 5 hertz conditioning, all of those muscles activate simultaneously, so we get enhanced excitability. There is a emerging kind of exciting model of true chronic pain where NGF, neural growth factor, is injected in a muscle, and it leads to increase in pain over the course of about 30 days that gradually resolves over time. When you do this injection and you TMS to find what activates the masseter muscle, there's a subset of individuals that have a depression of corticomotor excitability on that map, and some people that have facilitation, so the amount of tissue that can activate the muscle goes up. And so the facilitators have lower pain from the model, and the suppressors have higher pain. So people that have less excitability tend to have more pain. More recently, this has been combined with resting EEG and the peak alpha frequency, which I don't have time to talk about in detail, but peak alpha is emerging as a pain sensitivity measure. And it turns out when you use this NGF model and you combine peak alpha with corticomotor excitability, you have a perfect area under the curve, an area under the curve of 1, where you can predict high pain sensitive versus low pain sensitive individuals. And the key predictors are a lower peak alpha frequency. So the peak alpha band peak here is at a slightly lower frequency. And again, they see this facilitator versus depressor as a strong predictor. So this is really cool, but there's a lot of tissue in between the brain and the motor response. So this signal is modifiable by a variety of irrelevant, or at least irrelevant to us, activity or changes that may not be directly related to pain. And so the idea of stimulating and then measuring what happens in the brain at the same time is really compelling. So what we're doing now is TMS EEG, so electroencephalography with a TMS pulse, and we can see an evoked response. That response at certain intervals has positive deflections, negative deflections, and some of those are sensitive to different neuromodulators. So GABA-A blunts this initial negative deflection at 45 milliseconds after a TMS pulse. AMPA enhances this positive deflection. GABA-B enhances another negative deflection. So very specific functional and mechanistic associations with these fluctuations in activity. And in addition to excitability and pharmacology, we can start to understand beyond excitability, connectivity between brain regions. So when we stimulate one region, how does it propagate? and plasticity. So when we give conditioning pulses, how does it change over time? And notably, these differ between brain regions, but they're stable over time, which is great for a biomarker. We know that under conditions of acute experimental pain, that we see enhancement of that N45, the GABA A. So it's less excitable when you have more GABA. So that's what we're seeing there. And that also predicts pain. And so we know that primary motor cortex is a harbor of potential helpful biomarkers. Motor evoked potentials are helpful, particularly these cortical excitability measures. And then the N45 seems to be a very pain relevant GABAergic related measure that could be important in pain. But still, clinical population studies are limited. And so I've already run over here a little bit, but we've had the great fortune of partnering with a device company that allows us to stimulate a number of different brain regions across the brain with a standardized protocol that has a large data set of healthy adults with the exact same protocol. So we can just jump to doing pain mechanistic studies, get some resting EEG data, stimulate 6 different brain areas, and record the evoked response at all other brain areas. We have this built-in data set that tells us what a normal response looks like. And so we can compare our patients to the normal cohort, and then also understand over time and in different treatment situations how our patients' brain activity evolves. And so thanks to this device, which allows us to kind of skip the line in terms of the hard technical part of doing this work, we can start to measure excitability and plasticity in a more automated way. And so now we have a pipeline to do so. We've got some hypotheses about how to do this. We're going to implement some machine learning techniques. And in a preliminary feasibility study in a fibromyalgia population, we found, just as expected, that that N45 peak is reduced in individuals with fibromyalgia, indicating cortical hyperexcitability, less GABA tone. But we don't have any pain measures from these people. We don't know how it affects what medications they were on, a variety of limitations here. So we're going to now, I think the slide disappeared. So these TMS EEG measures are really exciting for us. And now we're moving into a new study where we're studying 50 patients with widespread pain, some with fibromyalgia, doing structured clinical interviews, quantitative sensory testing for acute pain, and resting EEG plus this TMS EEG protocol. And we're going to hopefully take a very noisy brain environment and get some clarity through stimulation. So thanks all for your time and attention. And I'll take any questions. Yeah.
So I may have not quite got to it, but so there's the idea that you have a peripheral increase in the muscle myotone, if you will, as a result of it.
Yes.
And you're taking that to be an increase in cortical excitability.
That's the term from the literature. I agree with you that that is not quite the right term to use to describe.
What I'm wondering is how do I know this is not a total spinal effect? Because I mean, the corticospinal we know is going to the motor neuron pool, but it does so to interneurons and so forth. And I can imagine that what you're actually doing is increasing the size of the receptive, not receptive field, but the, shall we say, field of excitation, which would then involve more of the myotone. I'm just being speculative.
No, I think that's a reasonable hypothesis. I don't have a good answer to that question because at least TMS only very recently has been translated back into animals. It's very difficult to get a focal enough field of stimulation to understand these sort of mechanisms. And TMS works differently from direct electrical stimulation, importantly. And it seems to have more of an effect on the intracortical GABAergic microcircuits. So it's a disinhibitory effect in some cases. And there's some studies looking at the effects of TMS on corticospinal tract spinal volleys and then on the resulting MEPs, but those have largely been done in non-human primates, and there isn't a large body of work on that. So I think that is a super interesting question. And part of the reason that I've moved away from MEPs as the outcome measure, the motor evoked part of that potential, is because there are all these unknowns between the cortex and the evoked motor response that are difficult for us to measure and to really get clarity on that. So the TMS EEG approach is using subthresholds. We're not getting motor evoked activity, but we are getting, importantly, corticocortical responses that we can measure with better resolution. And I think also understand a little bit more about the dynamics without having the influence of a very long track between brain and muscle. That's a really important question.
Can you talk a little bit more about the targets for pain and then the different targets for depression?
Yeah, great question. So last year I talked a little bit more about that because motor cortex was the first area studied with TMS because there's an observable motor response. So it's something that you can stimulate, you see a hand twitch, you know what you did is doing something, and you can see how doing other things changes that twitch. So motor cortex was first, and early studies showed that there was some pain benefit, and now there have been hundreds, if not thousands of studies showing pain benefit, but it's still not FDA approved because all those studies are kind of poorly done and heterogeneous. But depression's a different story. So it's left dorsolateral prefrontal cortex is the target. 5 centimeters anterior to M1 was the initial place that was studied. More recently, they tried to hone that in based on more individually tailored measurements. So like taking into account different skull sizes, and then even more recently using fMRI and connections between that area and other key regions in depression to better target to the DLPFC target. So that's the depression target. OCD is the other FDA approved indication, which is an anterior cingulate or an orbitofrontal target. And that uses a deep TMS system that shapes the field in such a way that it can penetrate deeper, but it's a little less specific as well. And notably, those require 4 to 6 weeks of every single day stimulation to get the clinical effect, which is still really impressive, but nobody's done that in pain. And so my study that I showed very briefly with a TMS clinical trial, 10 consecutive weekdays is among the longest that M1 TMS has been studied in a RCT type design. We're seeing better outcomes in the shorter studies. So there's something important about durability, and there's some principles across the mood indications, depression, OCD, about what frequency, what intensity, for how long, that are guiding us in the pain world to try to get better outcomes that are more akin to what has been seen in the depression literature. But the regions are different, importantly.
Do you think you'll be able to get to like weekly times 6 or times 8 because that every day?
Yeah, so the every day is very impractical. So I'm actually writing a grant to submit in June right now where we're going to do a multi-site trial of TMS. And we're going to do the term for it is suddenly escaping me, but it's among responders at the end of 2 weeks, we're going to do monthly booster sessions with either true or sham TMS to see if we can maintain durability over longer periods of time with just single booster once a month for a half hour. Which, at Stanford, they showed you could do 5 days of 10 sessions a day for 5 days and get a similar result to 6 weeks of everyday TMS. But during those 5 days, nobody else is going to be able to use that machine because 10 hours a day of TMS. So we're trying to find a way in the middle to get good results, good durability, but not have them come into clinic every day, all day to get stimulation.
Thanks. Great work.
Thank you.
All right. So Lawrence, I think I fixed the audio. That's my gift to you. Yeah, and I'm going to load up your presentation here. Well, I'll give you an introduction. I feel like it. Yeah, right. So next up, last and definitely not least, Lawrence Perret. He's clinical professor at UCSF. He's director of the neuromodulation program in anesthesia, and he was my mentor. Trained me 8 years ago, and still training. Taught me 60% of what I know, and so still teaching everybody how to implant spinal cord stimulators and treat patients. And so he is the current secretary of INS, as well as on the board of neuromodulation. He is president of the Institute of Neuromodulation and on the board of directors of NANS. I think founding member or founder of CalCIP and PSPS. And much more than that. But today he's going to be talking to you, and I'm really excited because, we all try to focus on successes and what works. And, we learn much more from failures. And so thanks.
Thank you very much, Prasad. That's quite the introduction there. So I know we're running a little bit behind, so I'm going to try to get going here pretty quickly. And so he gave the introduction, so I don't need to do that. What I want to talk about, though, is what is neuromodulation. We talked about that a little bit, but let's just go through that with some real overview here. What we've got here is technology at the neural interface. That neural interface can be peripheral nerves, spinal nerves, and the brain, right? And as part of that, we have, here we have brain stimulation with Dr. Eddie Chang, looking at the things that Prasad talked about earlier, with leads in the brain. We have Prasad looking at not only how to stimulate ideas, but also how to use external devices such as EEGs with Chad here to control our devices. So 2 different devices to control our devices. So at the essence of what we're doing, however, is the patient. We can't forget that that's the essence of what we're doing and why we're here. So the patient is the center, and by the term neuromodulation being the technology, we have to have the manufacturers, right? There's no getting around the fact that there is a device that's being made by someone, and those devices have to be regulated. Those regulations come from FDA and CMS, and they communicate with us physicians to tell us how to use it, when to use it, and why to use it. So that is the basic infrastructure of our neuromodulation ecosystem, with good communications from regulators to manufacturers, and the manufacturers back to the regulators, and both of them, of course, telling us physicians how to do it, when to do it, and why to do it. All right. In the last 25 years, we've seen a huge surge in technology. The manufacturers and the scientists have been just fantastic at providing us with greater technologies and stimulation parameters, and it's just been phenomenal. However, in spite of this increase, we do still have this problem with failure. And here we see that Van Boyten reported in 2017 a 20% failure in the course of 5 years for explants. And we go on to find out what those explants were for. Many times those explants were just because of lack of efficacy. We see here Pope reporting up to 44%, Van Boyten 20% in a couple of years, paddle lead 28%, and high frequency 22% in 2 years. So the question is, this is still an ongoing issue. I like this particular study with Dr. Roslow's group out of Oregon, looking at DBS, VNS, and SES. And when we look at these in a little bit more detail, we see that over the course of time, the dark line at the top is the maintenance of the therapy. And we can see for DBS and VNS, the therapy maintained over 10 years. However, for spinal cord stimulation, we see this huge dip in the therapy starting off within the first 2 years. But then we look at the devices. Yes, we expect the battery to wear out in a few years. And in fact, in DBS and VNS, you start to see some wearing out of the batteries in the 7, 8, 9 year period. But in SES, we're still seeing at 24 months, over 25% of the device is already failing. And that's just the device itself, all right? Here, Samuel Dawi's work with DRG reported a 69% related complications over the course of 24 months. One of the things that he mentions in the paper, however, is that the therapy for DRG is actually maintained. So even though the device is failing, you replace the device and then you get ongoing therapy. So it's really good for the therapy. The device has got another issue. Let's compare this to cardiac. When was the last time somebody told you, yeah, I got a 20% chance my pacemaker is not going to work in a couple of years? Would you put a pacemaker in? I don't think so. Here we see that 91% of the time, explant for pacemakers is due to expected battery depletion. So just normal known things, not this unexpected lack of efficacy. So what is the issue here? We've got great communication with the regulators and FDA manufacturers, great relationship with them telling us how and when to do it. But the question is, what else is going on here? I see a number of neuromodulators in here. So I got a quick question for you. Raise your hand if you have seen a device failure of any sort. Lead fracture, lead migration, battery problem. All right, so we got a number of people out here. All right, keep your hand up if you have personally submitted a device report to the FDA. You reported to the FDA. We got 3. All right, good, very good. Best response I've seen in any audience I've presented this to. Usually the case is people haven't done that.
What we've got here is failure to communicate.
That's right, failure to communicate. That's been the ongoing problem all along. What we have here is we as physicians and facilities have not been reliably sending that information back to the people who actually need to hear that information, the regulators and the manufacturers. In fact, the FDA actually issued guidance on this back in 1996. They said there is mandatory that we actually report to the FDA any actual deaths. But anything short of death can't be mandated by the FDA because they don't have any jurisdiction over facilities. So therefore they said, do what we think is good. And they gave some ideas. They said here, permit access to return devices to the manufacturer for evaluation. And you should report things like failures of efficacy, failures of leads, migration, all those things should be reported, but not mandated. Well, that's kind of the problem, right? Here we have the 3500 form from the FDA that you're supposed to use for reporting back. And we can see here that this particular form expires in 6/30/2025, and there's probably going to be some change in this form because this form talks about gender, and apparently gender is not a concept in our current administration. So when they advise the revision of this document, they'll probably remove that, which will be more data that we don't have. But nonetheless, not a lot of folks are getting that information. So as a result of that, as part of the Institute of Neuromodulation, a nonprofit charitable research subsidiary of the North American Neuromodulation Society, we conducted a survey of the NANS membership. So we took 1,400 of our members, sent them out surveys, didn't get the greatest response back. We got 104 back. And this was published, and the lead author is Dr. Tess Buthe, who was our incoming fellow for this next year. She's out in Stanford now. But other UC folks include Dave Dickerson, a former fellow, and Dr. Dhruva, who is in our cardiology division, as well as myself. And this is what we were looking at. We see here that the majority of the responders are actually anesthesiologists, and most of them, almost 50% or so, were actually in private practice. And to my surprise, the vast majority had significant experience, over 10, almost close to 20, over 20 years experience in implanting 20 to 40 devices per year. But we found out that 25% of the facilities had no known documentation policy for release of devices. 26% of the respondents had no knowledge of whether their facility would release to the manufacturer or not. 64% didn't even know who the designated reporter should be. And you see here in the middle of number B there, was it allowed to be released to the manufacturer? There was a small section of those that said no. UCSF was one of those sections. All right. And so then I inquired, why is this? And our risk management attorney at the time, I would say, this is just ignorance is bliss. Is that the point of this? And she said, no, it increases our risk and our exposure. And she's absolutely right about that. But the issue here is if you're not transparent, then what are you hiding? And so to take the words of Mother Teresa here, honesty and transparency make you vulnerable. Be honest and transparent anyway. And that is exactly the case. Being transparent about failures is a problem. And the risk management attorney was correct. It does increase our vulnerability and our risk. But, we can either be like those in charge who are not transparent, or we can be like Mother Teresa, your choice. So here we have an example of this, and an example that Tony Yax is very familiar with. And he was on, not this one, but the next paper I'll show you. But here we have intrathecal therapy. And this is Dr. North, who's on the Institute of Neuromodulation with me. And he was presented with this case that he reported 3 months after starting intrathecal therapy at 100 milligrams of morphine a day, concentrations that were approved by the FDA at that point in time at 50 milligrams per mil. This patient developed paraplegia, and the question was why? There was no real clarity on why. There was a discussion about, was this a catheter? Was this a chemical issue? Was this a drug issue? Lots of questions. And you can see here that one of the authors on this was Dr. Yax. This was 2000. This was the very first polyanalgesic consensus conference trying to identify what are the issues related to this particular problem. Most importantly, you look at what's written here. Those who do intrathecal therapy will find this rather interesting. Drug pump compatibility and the effect of the pump for the drug dilutants and pH are important considerations for all intraspinal drugs. Morphine has never been associated with problems related to these factors, and extensive clinical experience suggests that solutions up to 50 milligrams per mil are tolerated by the pump and are acceptable for long-term clinical use. All right, I hear laughter in the audience of people who actually do this kind of work. All right, so what we have is the 2003 Medtronic Initiated Implantable Performance Registry. I think this is quite laudable for Medtronic to say this is a problem and we're going to start a registry to figure some of this out. And as a result of that registry, they went out and asked some experts like who? Tony Yaks, to figure out what's going on. And what Dr. Yaks did discover was that not only was it morphine the problem, it was the concentration and the delivery. And that's why his discussion earlier on flow rate was so critically important to everything we do in intrathecal therapy today. It was the most important thing in intrathecal therapy in the last 30 years, in my opinion, to understand how and why the concentration and the drug concentrations have a huge impact on the granulome you see over there in that far corner. So now we are utilizing this every day when we're deciding what kind of drugs and how to implement those drugs into therapy. And now we come up to the 2024, the latest PAC guidelines, which Tony is still a member of and still contributing to. I have some input on this as well. And yet, when we look at that guideline, we do have morphine on the list here, but morphine and zaconotide are the only two FDA-approved drugs for intrathecal therapy. Everything else you see on this list, which is done by almost every other academic center in the United States, and most, even the private world as well, are not FDA approved. So we are routinely doing things that are not FDA approved. And yet, when we know that granulation is a problem due to the morphine, which is FDA approved, what do we recommend as our consensus statement? Use lipophilic agents, because we believe there's less of a problem with lipophilic agents. And we know that that is not FDA approved. So we are intentionally doing things that are not FDA approved and recommending this therapy to everyone else out there doing this. Well, what happened? Granulomas may have gone down, but then we have problems with the devices themselves. Here we see the fact that these lipophilic agents permeate through the internal membranes of the tubes in the catheter, causing corrosion of the gears in the pump, shorting of the circuitry, and causing motor stalls and failure of the pump and withdrawals of the patients. The FDA came out and said, hey, look, maybe it's you should be concerned about using these things. In 2018, they had this safety notice saying, if it's not FDA approved, you might have more problems. That caused a huge stir in the neuromodulation community, but we still did it. And finally, they came up with a decision to ask Medtronic to revise the device itself. And so now our newer devices have new gears with new material, encapsulation of the circuitry, and therefore we're having far less problems with pump failure as a result of these non-FDA approved substances. This is the reason for transparency. If we hide it, we don't find the problems. If we don't find the problems, we don't find the solutions. Here we have a perfect example of not being afraid, not using fear to dictate your policies, but instead using transparency and honesty to say, yes, it's a problem. And we see a continuing problem, we change that policy. How are we going to fix it? Doing good engineering, good clinical practice. This is the way it's done. And therefore, our need to improve and advance our communication with our manufacturers and our FDA is not only important, it's actually critical for good patient outcome. And for the words of Ronald Reagan, transparency and communication are important, but trust but verify. And so that leads me off to the next section is we have to do a better job of surveillance of these issues. We know these things are going on, but we don't necessarily know how they're going on in our populations as a whole. We do have an ecosystem here. What part of the ecosystem do we represent? Clinicians, professional societies, and facilities all need to be part of that ecosystem and contribute to understanding what the issues are. And each of us can decide to contribute part of the problem or part of the solution. It's our choice. Now, here we are, the largest healthcare training organization in the entire United States. The University of California, 21 health schools, including medicine, dentistry, nursing, optometry, pharmacy, public health, and veterinary medicine, and 15,000 health science students and trainees and residents. If we are not the leaders in this, then who is? If we are not the ones providing that kind of clarity, who is? It should be us. We are the fourth largest economy in the world. If we can't afford it, who can? We need to do a better job. And so we are. We are now actually have a process in place where we actually have a tracking form that the attorneys wanted us to keep, but that tracking form allows us to identify the case and the problem and send that information back to the FDA. And we've been routinely doing that ever since this started. But there are those who say that surveillance is a waste of time. The registries are a waste of time. One of my colleagues on the NANS board said that registries are good for short-term identification of problems for multiple institutions, but that open-ended surveillance is expensive and it's not really all that needed. Well, I would argue that that would be the case if we had good ideas about what was coming to the future. Can anyone in here tell me what these devices are and how they relate to intrathecal therapy? Exactly. Same thing I thought when I first heard about this. An iPhone and a charging cord. What do they have to do with intrathecal therapy? Well, it turns out they can both stop the pumps and cause patients to go into withdrawals. I first experienced this at Parnassus, right here at UCSF. In the middle of the night, patients were going into withdrawals. I could not figure out what was going on. I called Medtronic 800 number at 11:30 at night, and they told me to do something that I thought was crazy. After going through all the iterations and trying to interrogate, and I couldn't interrogate, they told me to do something very bizarre. The person on the other end of the phone says, lift up the covers. I said, what? Lift up the covers. And I said, well, why would I lift up the covers? Just go ahead and lift up the covers. I lifted up the covers, and that's what I saw. He said, oh yeah, that's your problem right there. Move it. Lo and behold, I moved it. Pump started working again. I did not report that. I probably should, but someone did report intrathecal baclofen failure because a person went around with their iPhone in their fanny pack in the front where the pump was, interfering with the delivery. No one understood that. So as we get more patients with more devices, we've already talking about STEM and pumps and patients. We have multiple patients with 2 devices. And now we're having more and more devices that are not just mechanical in nature, but they're actually AI in nature. And they're physiological closed loop control systems, the precursor to the AI. But that is coming very, very, very quickly. And those devices are going to have lots and lots of programming issues that need to be sorted out. And in fact, when I look at this problem, this is another ION project currently looking at the implementation of physiological closed-loop controlled neuromodulation devices. This is a primer for engineers and clinicians to understand how we both have to talk to one another and plan how to better manage our patients and our devices. This is being led by our colleagues out of Oxford, Drs. Victoria Marks and Tim Dennison, 2 engineers there. And when you look at the failures that we're having, this isn't just a simple mechanical failures anymore. We're starting to have here, Minnesota Star Tribune reports that there are 23 serious medical device recalls in 2 years. This is 2022 from 2020 to 2022 from Medtronic. But I'm looking just in the last year alone. And the one device that I use the most, which is intrathecal therapy, I've had 3 recalls. 1, 2, 3. Out of these 3 recalls or urgent field safety notices, 2 out of those 3 have been software issues. Software issues where the pumps are failing for a variety of reasons. We've got to understand that as we get more advanced technology with more physiological closed loop computerized controls issues, we're going to see more of these problems. All right. Now, what about surveillance? In order to surveil, you have to know what you're talking about. You have to have an identification. Everyone here has been in a vehicle, in a car of some sort. Every single one of those vehicles has a VIN number on it. Every single one of those VIN numbers can be tracked to an origin, manufacturer, make, model, everything about that. All right. We're putting devices into people who are running around the world and we don't know who, when, where, and why they got them. Not too sure about that. So the FDA issued this guidance in 2022, and this was the culmination of years of work that they were looking at to identify and place unified device identifiers, unique device identifiers. This has been in place since 2007 with the FDA Amendments Act. And you can see here that up to the present day, it is now mandatory mandatory for all implanted devices to have a unique device identifier that tells you everything you need to know, including the serial number, the production identifier, the location of things, and that is identifier that should be tracked and utilized by all of us. This is the one I placed in Monday. It's got a QR code. We have a scanner. Our ORs that I'm using are almost brand new. They're less than a year old. The whole building's less than a year old. We have the latest technology in our ORs at our Bayfront Surgery Center and our pain center. Scanners did not work. Scanners are not programmed to pick up any of this information. All right? We need to do better. What can the utilization of this do for us? Clinical workflow. The OR nurses who use this in hospitals that use this say they much better able to accurately place that information into the patient chart just by using the scanner. That does work. Better able to keep track of patients in adverse event reporting. Better ability to generate real world evidence. Better recall management. We can identify finance and revenue at institutions that use this say that their revenue's gone up significantly because they can track better. It turns out that many centers will bill a spinal cord stimulator and they can get the old fashioned version or they can get the new version. It's like billing the same amount for your Yugo versus your Ferrari. I don't think so. You need to know exactly what you're doing. All right. Inventory management and procurement. It turns out that the only people in the state of California currently using UDI is Dr. Cherry's organization. Hello, Dr. Cherry. It turns out that Kaiser is using this to track their devices in the same fashion as all the other GS1 issuing agencies, which includes all of Europe, China, Egypt, Saudi Arabia, Singapore, South Korea, Taiwan, Turkey, and the US. What other device or what other mechanism can you find that kind of broad utilization? Okay. But here in California, the only ones currently using this is Kaiser. All right. Now, how do you do this? Is it going to be complicated? Yes, it is. But guess what? Guess who actually developed the playbook for the utilization of UDI systems? Part of the team that was involved in that is Dr. Druva here at UCSF. So currently we have the playbook, which is entitled, is definitely complicated because we have to have our leadership engaged. We have to have the physicians engaged, our clinical staff, the supply chain, the IT people have to be involved. But if not here, then where? Certainly this is complicated, but we have all the tools necessary. We have all the expertise necessary. And so this reminds me of a phrase that I used to hear often from one of my thesis advisors and one of my mentors, someone who I highly respect, who is no longer with us.
With just a little phrase that goes for the brain as well as for muscles. You use it or you lose it. Use it or lose it.
Dr. Diamond, use it or lose it.
That's essentially what we believe. With use, you grow branches. With disuse, sitting around doing nothing, you lose it.
So we can either sit around and do nothing, or we can use all the resources we have to actually improve the quality of care of our patients, do better tracking, do better science, do better research, and have a better outcome for all of our patients and help develop the next future generation of neuromodulation. Thank you very much for your attention. And for those who want to explore this a little bit more, you're welcome to attend the Istanbul European Neuromodulation Society meeting in a couple of weeks in Istanbul, where they'll be utilizing a new system where they're taking all the various countries that have all of their registries for neuromodulation and combining them into one European neuromodulation registry. And if you're really interested, we can talk about this more in Kazakhstan at Almaty meeting later in the fall, and then again next May in Portugal at the International Neuromodulation Society. Thank you very much. I'll take any questions, but while I take any questions, I want all the speakers to come back up to the front and we can all share in questions and answers. So please, all speakers come back up. Any, we can just stand. Yeah, any questions about this particular component. Eric.
First of all, thanks very much for taking on this accountability project in I'll say in a broad way. It's not glamorous, but it's essential, like anything. But I want to back up to this question of explants, and I want to give. I guess I have a positive and a challenge for implanters based on what I see. There's obviously a higher rate of explant with spinal cord stimulation compared to DBS, for example. But I'm not sure that's an indictment of the therapy as much as it is the ecosystem that goes around its placement. For example, the front end of placing a DBS device is a little heavy. It takes a couple of holes in your head, and that's done by only a small number of individuals, physicians, and it's done almost always in a university setting, where interestingly, there's no particular financial incentive for that individual to do more surgery or less. A majority of spinal cord stimulation devices are placed in the private community, largely with physicians who have interest in a surgery center, and it's reasonably profitable, simple, low morbidity procedure. So the front end is much easier with a spinal cord stimulation device than a DBS, and it's there's positive reinforcement for the surgeon to do more of them. Well, we also don't know how many of the explants are actually being, of the continued use of DBS means continued device presence in the body, right, with
Yes.
DBS. It doesn't necessarily mean active use. And on the back end, the hurdle for taking a DBS device out is significant. For taking a spinal cord stimulation device out, it's 15 minutes in a local anesthesia and a go-home procedure. So there's issues there, and I want to say that I don't mean for it to be an indictment of my colleagues, but there's a powerful incentive to place the devices that's easy and safe on the spinal cord stimulation side. And then on the DBS side, if they're not used, they may just stay in there, and so they don't get registered as an explant. So it's a complicated bit of data, but again, I'll just applaud you for taking on this challenge, because I drafted the ISPR protocol for Medtronic 20 years ago, and was involved in its implementation and so on. So we obviously, and they did a good job of collecting the data. They also have an incentive because they use that with FDA, to achieve the changes without having to do clinical trials and so on. But continued pressure on accountability is going to be more and more needed as we go forward, because we're going to have fewer and fewer people, frankly, trained in surgery doing the implants. And that's just a reality of non-anesthesiologists and non-surgeons doing more and more implantation. So long-winded, thank you, good job, and I'm in for whatever we can do to continue the tracking and accountability.
Thank you very much. And I was, after being in private practice for 15 years and then joining UC and being here for the last 10 years, what shocked me about my practice. And one day it just dawned on me, I said, I've got to go look at the data. I can't believe my sense without doing way too many explants. What's going on here? And I went and looked at the data and found out that something like 60% of all my cases were explants. And when I looked at where they're coming from, they weren't people who were implanted. And so when I called the company and said, hey, we got a case coming up as an explant. I would first ask them, how are they doing? Oh, they're doing great. I said, well, it's coming out in a couple of days, but they're saying it's not doing so great. But I first thought, was this just a couple of bad actors who are implanting? And then I looked across the board and people I know, and the patients say, yeah, I had this issue or that issue, and it seemed to be working out well, but then later on, it didn't. There's a number of issues. And the whole reason we need a registry and surveillance is because I am no longer convinced that I can say it's simply this or that. I think we need real data to really characterize it, but the numbers are certainly there. We have a huge number of explants at UCSF, and they come from all over the country, and even outside the country at times. And we just need to do better, but I can't do better if I can't track it back to when it got implanted. That's why I need a device identifier to track it back to where it started from, to where if I'm taking it out, it's the final outcome of that device. We need to understand everything that happened in between, but it's a bit of a challenge, but we're not going to get there if we don't get the data. Yeah, Elyssa.
Well, I guess I agree with Dr. Grigsby that about the whole idea of incentives are a little out of whack. And one of the other things that has occurred is with the studies showing, oh, if you take this one out and you put in a new one, then that, you know, we can salvage it. And so then there's more incentive for taking out and putting in. But you know that, so just a point there. About the FDA reporting, very difficult to do. We do it, but I did want to ask a question. We also have promises from the companies when we return them to them that they're reporting. And I wonder, I have trusted that because it's been easier to do it that way. And so we send them back to the company, they pick them up and we report what the problem is. Is that actually happening, that they're reporting to be doing good reporting, you think?
So certainly is mandated by their agreement with the FDA, but I actually make sure for everyone that we put in through the university that we expect in a reasonable amount of time to have a report back from the company about whatever it is that we're reporting to them, and then they're giving us a report back, and that report is actually going to the FDA. But do I think it's happening 100% of the time? I know for a fact that it's not, but it's supposed to be. But once again, if we have a registry and we're tracking these things, then we can say, for example, I sent you X number of devices, I got 3 reports back, and I sent you 6, where are the other 2? And let's see the tracking of the FDA. So it's not being monitored because the FDA doesn't have jurisdiction over facilities. And so they do have jurisdiction over the manufacturer, however. So if we report to the FDA that we have seen this problem with a report, then the FDA then is alerted, hey, there's been this problem, there's been a report to the company. And so they're expecting to see that report, but it could be better. But what's the data? It's not been easy to keep track of. So until we can keep track of it, we won't be able to see how well we're getting this complete closure of the loop. That's why it's called a closed loop of failures, because we all have to communicate on an ongoing basis.
There's a question by Tori Marks.
Oh, great.
What can other institutions learn from the experiences of the University of California system? And the follow-up question is there a map they can use to set up similar infrastructures at their institution?
Well, thanks a lot for joining, Tori. She's in Oxford right now, and so it's a bit late for her in the evening. So Tori, I'd say that what we're learning is that there's plenty of room for opportunity for growth and development. That's what you can learn from the University of California, and though that we can move forward in that, as we did with the reporting issue. And as you saw that the UDI was I highly recommend that all institutions look at the playbook. I just showed you a couple of pieces of it, but it's a very good playbook on how to implement UDI, the universal device identifier. These are the things that we need to start with and go forward with. You know, Tori, when you are seeing what's happening in your region of the world in the UK actually has the best spinal cord stimulation registry in the world, in my opinion. It's the one that the whole European Union is starting to model after. And as a result of their registry, they were able to actually identify that spinal cord stimulation only sat a couple of steps below hip replacement as a very impactful procedure for quality of life changes. They only able to get that data by having a registry and managing. So actually what's happening, I would turn your question around, Tori, and say, I'm looking to the work in the UK as a guide to how we should be doing things better here.
We have about 4 minutes left, and I'll maybe just ask one question to all fellow speakers. Starting with you, Tony, what are you looking most forward to in the coming year regarding, you know, neuromodulation or intrathecal therapy in general? Like, what maybe experiments, what clinical therapy, what data, what are you most looking forward to?
There's all sorts of things being developed with regard to the role that goes through ganglia. And I'll just say that I think that's an exciting area. And then, of course, the other issue. But I thought the speakers really brought up a lot of, I've learned a great deal, particularly about the non-invasive imaging side of the equation. I personally looking more forward to that.
I'm looking forward to UCSF, not UCSF, the University of California, all the UC campuses combining in a surveillance registry. That's really what I would love to see.
Well, I'll go next. I'm honestly looking really forward to following up on what Tony said. How non-invasive technologies can be used to image, right? Obviously in animals, but also to stimulate, to neuromodulate. And so right now, and there's a trend, when people think about deep brain stimulation or drilling holes in the skull, like you mentioned, Eric, there's a high barrier to entry, right? And how can we do this? How can we target structures or even conduct experiments in animals without having to do all these laborious things, expensive things, in reproducible manner? And so right now, I think, this, there's so much money in the space that's being thrown to developing functional ultrasound for brain stimulation for humans. And I would argue, it's another example of technology being ahead of the science. We don't even know what we're doing. So yeah, that's what I'll say.
I agree.
Looking forward to similar things, although I think the main thing that I've been very interested in is precision medicine for pain. I think we're getting to a point where we're able, from an imaging standpoint, peripheral biomarker standpoint, deep phenotyping enabled by AI, getting to a point where cancer was, 15 years ago, in terms of our ability to identify mechanistic biomarkers that are unique to individuals and then treat to those biomarkers. So I'm hopeful for that.
Yeah, so I speak more from the kind of more basic science and technological standpoint. I'm not a clinician. And yeah, we're spending a lot of effort right now, as Prasad brought up, to develop these technologies for non-invasive neuromodulation based on ultrasound. But it is very interesting to see that we have some, I wouldn't call it easier issues, but that don't need necessarily like a whole new fancy technology to bring a whole new fancy technology to the table. And there are many issues that can be solved. And I think what is important for us as people developing this technology, I think it is important to keep that perspective into account, right? We can come up with new devices with integrated AI and closed loop and so on. But then, yeah, I think it is important to keep an eye on kind of some of those more kind of, I would say, tangible, almost like issues, like having an identification number and so on. So I think it was very interesting to hear your perspective. So, thanks.
I agree with all of those comments, but just to say something a little bit different, I think on the pharmacology side, we're finally getting to a place where we're starting to understand even the opioid receptors at a level where we should be able to pharmacologically pick out pain relief versus addiction, maybe not in the next 12 months, but maybe in the next 5 years. And so I'm very excited about some of the things that we're learning on that side as well, where we could still use oral therapeutics to help people.
Any other questions or comments? Well, I'd like to thank you all for attending. We're going to have lunch and a networking session out here, and I think Dr. Shirvalkar would like to get some comments about PARC in general. Thank you.
So first off, I want to thank everyone for attending. Also, let's a round of applause for take off early, but again, so we attempted to create Q&A and networking, but hopefully you can stay for the next hour or whenever, but we have lunch for everybody out there. I think the networking is equally as important as all the talks and such. We're very excited to keep all this going. Again, I also want to recognize Anna Landau, who this meeting would not occur without her. Prasad and Lawrence and all. So thanks again, and hopefully we'll see you again in a year's time. So thank you.